Okay, thanks. So, okay. So, we are going into the next lesson. I think I just realized that we are supposed to speak about verbal hypothesis and other things in translation. A few things are there. I'll make up for them in some class, but they're all simple concepts anyway. Trying to explain the molecular basis of degeneracy in the genetic code. We'll get to that later. Right now what we are going to look at is the regulation in prokaryotes, gene regulation in prokaryotes. We have prokaryotes, and then we will discuss something that's more complex, that's in eukaryotes. I hope you have watched the videos and understood a little bit at least. And in this particular chapter we have some very important things. There are lacoperon, tripoperon, lambda phage. I will not go into as much depth as it is in the videos. We will go with confine ourselves to the syllabus at this point. If there is time or if we have enough things, we will always look at it. But otherwise, you have the videos for detailed things about lambda phage and other things. But we'll see how far we get. So, if we know, we should know the importance of regulation, right? Everything is regulation. Everywhere we use it in our daily lives, and every organism uses it in daily life. Every um, you can take every cell also uses that way, because if there is an entity and this is the rest of all that is surrounding it is the universe, and the universe is constantly changing, right? It's not that it has a brain or something that it wants to change. It just happens. Change is the norm. Being stable is the abnormality, right? That is how we should see. Change is the nature of a natural course of everything. So here we are seeing an entity, and the environment is changing. Say for example you have a rock, and that rock over periods of time, because of changes in its surroundings, will keep changing itself. Not that it is doing it as a conscious exercise, it just get changed because of the wind or water. It keeps eroding and becoming into smaller and degrading. You can take moon for example. Moon is in the space. It's almost within its physical entity. As a physical entity, it's not in contact with any physical entity, any other physical entity. It is still influenced by the cosmic dust or stones, asteroids, and then there keeps pits and other things keep forming. So if you think of it in a philosophical way, the whole environment is changing, and that change is going to influence the every entity in this universe. Some entities will take may change rapidly, some may change very gradually, and sometimes they might take millions or billions of years. That is beyond our perception or our ability to observe and record. So for there is something with animals, or there is something with living organisms, that is, if there is a change from outside, and if the entity here that we are talking about can make a compensatory change within itself, so that it can adapt and exist in that changing environment as well. That's sorry for these kinds of disturbances. Okay, so the difference between living organisms and non-living things is that non-living things are at the mercy of the changes that happen in their surroundings. If you see living organisms, they have some inherent capacity to exist in time despite the changes that happen in the surroundings. Okay, and that ability to change itself is the essence of it is from the um, because of regulation, and that regulation is about gene regulation, right? So I hope you can see it this way. Take a rock and take a, a dog. A rock over a period of time will not be able to change. It is only going downwards. It will keep degrading and degrading. You can have discussions or counteract what do you say, counter arguments against it. Say some iron piece is getting rusted and it's growing. You can also talk of magnets. They have different properties, inherent properties. Magnetism is an inherent property, right? So that all comes to at a later time. Maybe we will discuss if possible. But I want you to know that all the adaptations, all the changes in organisms, everything is because of change in the nature of gene expression. And that is what we are going to discuss in regulation. So every organism has genetic information. Every The genetic information of all organisms are vulnerable to changes, that is mutation, and every genetic information has has the scope for modulation, that is expression, repression in different times, and all those things come under regulation. In this chapter, lacoperon and tryptopherans are very important, and also lambda phage, right? Those are basic concepts. The others are a little bit advanced, but that's all right. We'll have a good enough introduction to them, and that's considerably enough. About sigma factors, we already learned a little bit when we were reading about translation. But let's get to this. All organisms, all species are in competition. The amount of resources are limited, and the competition is heavy, right? So, and also as one cell or one organisms keeps dividing into two, and those two will become more, I mean, will keep reproducing, and they'll keep doubling each time. And they're doing that. Say for example, this is all the nutrition that is available. They are already consuming it and becoming more in number. More in number would require more amount of food, but the resource is limited, right? So all the organisms are in a race for who is able to consume more and live longer or outnumber. There are two things, spatial and temporal. Spatial, you can say, who is able to leave more number of offspring is one way, spatially, occupying space. The next one is who is going to live longer, that is temporal, in time scale. These are, if you again, spatial is also increase in spatial or increasing number of copies is also important for surviving longer. For that species to exist longer, it has to reproduce more, or it should be very tolerant to stress. So life is all about efficiency, that the more efficient ones, they will survive, the lesser ones, they will die out. So it is about speed and accuracy, whether it is correct or not, and how many demands are there for survival of that organism. So you have to, I mean, there are some things that we can always imbibe into our daily life about regulation and seeing. We try to do, if we plan to do, we don't want to do any mistake at all. The only way is to not to do anything at all. And if you want to do it very very fast, you're going to do a lot of things and they're all useless. And if you say, I want to have a car to go to work, I want an aeroplane to go to work, and if you're trying to have more and more demands for every small work that you're doing, then it is also a burden. So we have to, the highest quality is perfectionism, and perfectionism is a kind of illusion, because on, if you meet if you meet the target, that is when it is perfect. That's perfect or good, right? So in the given time, within the resources available, did you finish the job or not, is what's the question. And that's the same case with every living organism. Within the available time, did you grow and reproduce or not? That's how organisms, or all living organisms, their race is that try to find food, somehow reproduce and give birth to healthy offspring, so that they can recapitulate what you have done, what the parent has done. That is how this cycle of generations in organisms also happen. Okay, so when we talk about gene regulation, every event that you can think of is associated with gene regulation. And we know the central dogma: replication. You can think of transcription, translation, and then protein stability. You can take any of these things, and all of them are good sources for regulation, gene regulation. So initiation of transcription, we have learned a lot of these things, especially in prokaryotes. The one that I'm showing here is that for a eukaryote. So eukaryotes also have an additional one, something like exon splicing, right? And we have learned about alternate splicing, and we have taken up one or two examples on how regulatable alternative splicing can happen. Then we also have which ones can cross over the through the plasma nuclear membrane, and which ones get translated, and how much should they get translated. There is also, that is about regulating translation, that we'll see in this lesson, and about the stability of mRNA. If the mRNA is stable, that means exists for longer, it will give rise to more number of proteins upon translation. But if the mRNA is degraded faster, then the proteins that are formed are actually lesser, right? So mRNA, stability of mRNA is one way of regulating gene expression, and protein synthesis is one. And then you can also have post-translational modifications, something like phosphorylation, like all the histone modifications that we have learned, and so on. What I want you to understand about gene regulation is, okay, now here is the gene, okay, and ultimately all the proteins or enzymes, could be any of them, or RNA even, they will give rise to a phenotype, right? A phenotype is a characteristic or trait. Okay, I can't write with the mouse, but yes, I hope you understood. The gene is here, and it has to result in a trait. And the trait can also be like, say for example, skin color. It could be 10% skin color dark, or 50%, or 100% dark. How can that happen? And all the things in between the gene, remember this: gene by itself is useless. It does not give rise to a trait. It has to be expressed, and the so expression would involve transcription, translation, and so on, and protein and post translational modification, and also protein localization. The protein that is produced should be sent to the particular destination. If it has to be secreted out of the cell, it should be secreted, otherwise it's not useful. So from gene to the formation of a trait, a phenotype, every activity in between are prone to regulation, right? So you can talk about transcription, processing, or gene preparation. There are also there silencing and other things we will discuss later. Um, say for example, we learned about genomic rearrangements. I'm just taking simple example of hin recombinase. We spoke about the flagellin A or flagellin B, whether the flagella of that particular bacterium is made up of. We saw how transposition or site specific recombination could change a flagellum made of flagellin A to flagellin B and vice versa. We have also seen mating type switching that happens in yeast, mat locus. They are that is also gene regulation at the level of genomic rearrangements. And many of the things that are described here will be uh, in this place we will describe them in epigenetics, and some we have already seen. We will see them in epigenetics and promoter transcription and processing, RNA export, RNA turnover, that is the stability of RNA, mRNA, protein synthesis, protein modification, protein targeting, that is the localization, whether it should go to a lysosome or it should go to Golgi complex or endoplasmic reticulum or extracellular site, and also the protein turnover, whether a protein is stable or degraded immediately. Okay. The simplest one, what we have discussed as the introduction part, the pictures that I have shown are for typically for eukaryotes, but this lesson is about prokaryotes, and those are the best understood ones. And it's ingenious how a simple bacterium can respond to the environmental stresses, availability of resources, and so on. So the simplest way, if you look at, it's all about regulators, activators, and repressors. These are the simplest way you can think of. There is a promoter, and the promoter has the elements -10 and -35. And what may happen is there is also a site called as operator, and there is another site called as activator binding site. So repressors bind to operator regions. You call it operator because they are like switch on, switch off kind of sense they feel that we can think of. So typically a promoter, it all depends on the design of the promoter and many other things. Okay. So if the promoter, within the promoter, you may have different elements. You have the promoter elements like -10, -30, up site, and so on. But there could also be other genetic elements like activator binding site or an operator region. And where these operator regions are located will matter. How much do they overlap with the promoter elements will matter, right? As you can see in figure B here, in this case, the repressor is bound to the operator region, so there is no transcription. Why is that? Because the repressor is occupying the space of the promoter where the sigma factor or along with RNA polymerase should be binding. The repressor is obstructing the binding or the interaction of the sigma factor with that of the promoter elements. So will not be any transcription. In other cases, the promoter element that is shown here might be weak, and it might need some additional factors like activators to bind here, and this activator will allow the binding of or will enhance the binding of RNA polymerase to the promoter site. It is in contrast to the activity of repressor. Repressor is obstructing the binding of RNA polymerase, whereas activator is enhancing the interaction of RNA polymerase with the promoter. So in this case, you will have a very high level of transcription. We should understand what is basal level. Basal level is the minimal amount of transcription that would have occurred when no other protein factors are bound to the promoter elements. Only promoter is present as such, and you have RNA polymerase and sigma factor. No activators, no repressors. At that time also you will have some transcription very low level. But if repressor binds, that basal level is also not expressed, and it is completely shut down, kind of. Okay, so that is how we should see these things. And there are also some genes which are constitutive expression. That means they're constantly expressed. They will keep on producing mRNA. Those mRNA will keep on producing proteins. Those proteins will perform an enzymatic activity. Say, for example, glucose, those enzymes involved in glycolysis. All cells need ATP, and ATP can be produced continuously without the requirement of oxygen by glycolysis. Glycolysis is an anaerobic reaction. Not that it should happen only in anaerobes and not in aerobes, but the point is that glycolysis does not require oxygen, right? That's why you just refer it to as anaerobic cycle or reaction cycle. So in that, if a cell needs to produce this energy consistently, then you would want glycolysis to happen consistently or constantly, and for that you will need the expression of enzymes from those open genes. So you can, at least one of the examples is for constitutive expression is you can think of glycolysis things, and in eukaryotes or animal cells, beta actin, I guess, there are some genes that are constitutively active. They are expressed all the time. Basal level I described. Operator, that is the site where the negative regulator or the repressor binds. And recruitment is something like activator binds, activator recruits the RNA polymerase to the promoter elements. And that is also cooperative binding. Do you remember in biochemistry, you must have read about hemoglobin, and you know what is cooperativity. That is, if one subunit binds oxygen, all of them become an open conformation or so, and they all will accept oxygen atom. That is, binding in one unit is changing the conformational state of the other ones, because of which they also accept oxygen. In a similar way, binding of one factor here could enhance the binding of other ones. This could be by specific interactions, and this could be by the conformational states that happen because there is a change in one subunit, right? Allosteric is by binding of a ligand like element. Okay, I think we have enough background of what some of the terminology, so we can actually discuss some more factors. So the DNA that we learn, we should never see it as a stable, structurally rigid one. We should rather see it in lengthwise, because the genome is long. It actually bends and folds to a very great degree. Very distant elements or in the DNA can come close by like this, and their interaction could result in some consequences of the gene expression. And the interaction typically is enhanced by proteins that bind to those sites. So if they are like this, they can if the A and B could be binding, and then they could either recruit a repressor or recruit a RNA polymerase or other factors. It all depends on the affinities or specificities of the proteins that we are talking about. What we are seeing here is the point that we need to understand is that the DNA can bend, and there are interactions very far off. Genetic elements can actually come in close proximity by bending of the DNA. And there are proteins, something like a DNA bending protein. We learned about IHF. Um, IHF is involved in the interaction of or the prophage. In site-specific recombination, we learned that lambda phage integrates into bacterial genome. In that, we learned about IHF, integration host factor, I think. So that binding of which will bend the DNA like this, and it will bring the activator, I mean, the activator binding site to which the activator will bind, and the activator will allow the recruitment or activation of the RNA polymerase to enhance the transcription of the gene downstream. Okay. So before we go further, there are a few more things that we will learn, and then we will see how far we get. Negative regulation and positive regulation. So in both these cases, the point about these things is, in negative regulation we are talking about repressor. In positive regulation we are talking about activator. Okay, so anytime you hear the word negative regulation, that means it is associated with we are talking about an operon and a repressor. So we'll just look at this briefly and then we'll see. So in negative regulation, what may happen is, okay, this is the promoter that you're seeing here, and operator is the region where the repressor binds. Here we have a repressor that is bound here. So if that repressor is bound, what will happen? Gene expression will not happen. But if gene expression should happen here, there may be a signal molecule, something like a ligand, for example. If it binds to the repressor, then the repressor will not inhibit, will not have affinity for the um, operator region, so it will not bind. If the repressor is not bound here anymore, you will have transcription, right? So that is negative. And whether it is about we'll talk about the signal molecule, effects of signal molecule in a while. Just let's finish another way. These what I'm seeing in all these A, B, C, D, all these scenarios, we are having general talk. It's not a specific example. It's about how it is described and the variety of regulations that are usually available to us or available in the genes. In another case, like in the B, what you're seeing is repressor is only active. When I say repressor active, repressor has the ability to bind to the operator region. So in case A, like you're seeing here, the repressor is active if it is not bound by the signal molecule. If it is bound by the signal molecule, it cannot bind to the operator region anymore. In B, the repressor is active only if the signal molecule is bound to the repressor, right? I mean, the signal molecule or the repressor is active when it is bound with the signal molecule, and then it represses the transcription. So these two are the common between or the similarities between A and B is that the regulation is dependent on a repressor. In the first case, the presence of signal molecule will inactivate the repressor. In case B, the presence of signal molecule will activate the repressor and ensure um, in repression of the genes. We'll also look at positive regulation. We'll come back to these maybe in 2 days, maybe today and also in the next class, but because that is the general theme in most of the regulations. In positive regulation, we are talking about activator. If you're talking about activator, we will also talk about the DNA element or genetic element where activator binds is called as activator binding site and so on. So here also we have a signal molecule. Okay, but we will talk about again, in this way, in this scenario, in positive regulation, active, when you say active activator, then it means that it can interact with the activator binding site. If you're saying the activator is inactive, which would mean that the activator is unable to bind to the activator binding site. So in C, if you see the activator is bound to the activator binding site, so it can recruit RNA polymerase and mRNA can be produced. But if there is a regulatory element, a molecule that interacts with the activator, then the activator cannot bind to the activator binding site and repression would occur. I will repeat this again. So activator here in C, in this one, it gets inactivated by binding of the signal molecule. So presence of signal molecule will result in non-expression or repression of gene expression here. Okay. In D, what is happening? The activator is only in active state or active conformation if only it is bound by the signal molecule. Okay, some kind of another protein or small ligands and so on. It can be metabolic molecules also, the signals. Okay, in first one, in negative regulation is about repressor. In positive regulation, it is about activator. And here, negative inducible, the table that I'm showing here, negative inducible is what does the protein do? The action of the modulator. What is the modulator here? The signal molecule that we are talking about, the gray, this one. What is the activity of it? What are we saying? Inducible means allowing the inducible. When you say, that means it is always repressed. Okay, it is always repressed. That could be changed to expressed. By what is the default? That it is repressed. And what can be done? It can be allowed to be induced. And that is simply negative inducible. When you say default, it is repressed. That means the repressor is always bound here and it is repressing the expression of this gene. And the presence of a molecule will inactivate the repressor. Therefore, there will be induction of expression from this gene. So that's why you call it as negative inducible. These are terminologies that many people use. When you read a research article, for example, they simply say negative inducible, and it becomes so difficult for novice people like us to understand it. And that is why I'm trying to emphasize this, so you'll feel comfortable when you're reading research papers in the future. What is meant by negative repressible? By default, it is not repressed. It is about when you say negative, it is about repressor. Repressible means you're saying by default it's actually active and you are repressing it, and that is the activity of the modulator that we are, or the signal molecule. The product makes repressor active. Otherwise, the repressor by itself is in negative or in inactive state. And similarly, we have for positive inducible. That means these are the signal molecule will activate the activator, which is otherwise not active. The substrate makes activator active. And the other one is positive repressible. And that means it's about activator. And when you say repressible, the default state would have been induced, which can only be reduced, right? So we'll just, to finish this one part, we will describe what is A. It is negative because we are talking about a repressor, negative, and the repressor by itself is repressing, is in active state, and the binding of signal molecule is inactivating the repressor, so it is negative inducible. I'll just put as IN. Okay, negative inducible. What is C? It is positive because we're talking about activator, positive, and the activator by itself is able to activate, and this one is going to, the presence of the signal molecule or the modulator is going to inactivate. So it is, and if it is inactivated, then the expression would become lesser, so it is positive repressible. Okay, positive repressible. I'm trying to match the following in here. So A comes to this, and B, sorry, C is positive repressible because the presence of product makes activator inactive. And in B case, it is negative, and the repressor is active only in the presence of the signal molecule. So that is, otherwise, if the signal molecule is not present, it is active. The presence of signal molecule is actually making it into active repressor. So that is the negative repressible. And then D, it's about activator, and the presence, for the active state of activator, it needs the signal molecule. That means if this is not there, then the activator is non-functional. It does not bind here and allow the expression. So it would be otherwise repressible. So this is positive inducible. Okay. If you didn't get these all, it's all right. We can always discuss this in the next class as well. So you will be able to, it is sometimes confusing. Even if you ask, I might get confused sometimes all of a sudden if you ask. But simple thing is about trying to know positive and negative is easy. Positive is about activator, negative is about repressor. That much you remember for this time. So the lac-operon, I'll give a brief introduction to lac-operon, and I will try to discuss a few things that you will need to understand lac-operon in the next class, and I'll try to finish quite more in the next class. So the lac-operon, always remember that when you are writing about genes, you have to write in small letters like this, LAC. LAC is for lac, and it should be in italics. This is for prokaryotes, okay? But if I want to talk about lac protein, I'm just making up, then I should use normal. It should not be italics, and the first letter should be capital, and the next two letters will be smaller. So always try to know why it seems like an inconsistency or lack of something. It is not that case. There are standard ways of representing genes, proteins, and other things. We need to, it's like the syntax people use to tell. If somebody writes it as in italics, lac like this, that means they are referring to the genes. If they write like this, that means they are referring it to the protein that is produced of that gene. Okay. So here we have the +1, that is where the transcriptional start site, -10. You remember the -10 sequence? It's actually TAT, AAT. There are two things that are not matching, but it is still good enough considering that there is the spacer is good. Here also it is good. I hope you remember -35 sequence. If not, that's all right. The sequence is TTG ACA. TTG, this T is wrong, right? Here it has 3 T's, but otherwise TTG ACA is the -35 sequence. There are also one other site called as cap binding site. That is a protein, that's an activator that will bind here. And there is another site that is here, DNA covered by repressor, that is actually the operator region. Okay. From here to here, DNA polymerase binds. I hope you are able to, 1 second, sorry. Sorry. So if you're looking at the way the genome, the DNA elements are arranged, excuse me again. Yeah, sorry. So from here to here is the promoter region. You can think of it like that. That is the area that is covered by the RNA polymerase, and we have area covered by the repressor. Okay, or that is the area of the lac operator. If you see, lac operator starts from here to somewhere there, and it has a kind of you can see here that is the lac operator structure. You read this AATTGT GAGC. You can read the same thing here, AATTGT, right? This much is same until here and here. You're reading it this way. So there are little bit differences, but you see that there is a kind of symmetry between these sequences, right? These are referred to as palindromes. I think when we were learning about restriction enzymes, we did learn a little bit about palindromes as well. So the cap is the activator that binds to the DNA here. If the repressor is present, it will be bound here. And if repressor is not bound and cap is bound here, then DNA polymerase would bind, sorry, RNA polymerase would bind here and allow the transcription. You can have an equation. I'll just stop with that. Only DNA polymerase, only RNA polymerase is bound when it is basal level of expression. That means it's minimal, but there is some expression. Along with cap plus, if cap plus is also there, then you'll have very good expression of the gene. Transcription will happen well. But if repressor is bound, then there is no expression at all from this gene. Okay, I'll stop here.